Students, continuing with molecular biology techniques, we will cover up with certain topics which we've already discussed. But this class onwards, we will understand how to write these topics in a small answer or a short essay format. So, moving on to this kind of answering, so we'll go on to a question which can be asked, that is DNA libraries. Okay, so DNA library is nothing but any library as we go and see where there is collection of books, but here you have a collection of DNAs. So the DNA containing clones are stored or it is preserved as a source for a DNA. And there are two main types of DNA libraries. You have the genomic library and you have the cDNA library. So what do you mean by genomic library? It is a collection of different DNA clones representing the entire genome of the organism. So it is prepared by isolating the genomic DNA, breaking it into fragments of suitable length and then inserting each fragment into the vector which is cloned. And the collection of all these clones representing the entire genome constitutes the genomic library. So it also represents the entire genome, including both the coding and the non-coding region of the DNA. Okay, so this is the genomic DNA. Then the cDNA library, it is a collection of those DNA sequence or genes which are already expressed. So it is prepared from mRNA and it is obtained from specific cells of an organism. For example, the insulin beta cells of the pancreas are used. That is, the cDNA is prepared from the mRNA using reverse transcriptase, and then finally the double-stranded DNA is made, which is then used to make clones, and after inserting the DNA into the vectors, as in the case of a genomic library. So this cDNA library represents a collection of all the expressed genes of a cell or of an organism. So thus the genomic library and cDNA library are not the same. The genomic library clearly contains many sequences which are absent in the cDNA. And cDNA library from different cells may show significant variations. Example, the cDNA library from the reticulocyte shows many copies of the globulin gene which may not be seen in the pancreatic cell because pancreatic cells do not express the globulin gene. So how do you construct the cDNA library? The mRNAs from an organism, it is extracted and a complementary double-stranded transcriptase enzyme is attached. The resulting DNA fragments are then inserted into a suitable vector and clone and creates a population of a clone, and we call this as a cDNA library. Since there are no introns in the mRNA, the expression of the gene is much easier. So this is the way how to construct a cDNA library. So DNA library is asked as a short essay. So you'll have to write about both the genomic library and also the cDNA for this answer. So moving on to another short essay, that is DNA probes. So we've studied about DNA probes in many of the techniques. So we'll understand how to write a short note when DNA comes as a short answer. So DNA probes are radioactively labeled single-stranded DNA molecules that are able to specifically hybridize or annihilate to particular denatured DNA sequences. So there are different kinds of probes which has been recognized. So these are of three types. Okay, so this is the single gene probe. Then you have the allele specific oligonucleotides and you have highly repetitive sequence probe. So the single gene, it recognizes only a single gene and does not differentiate among the alleles. So it is helpful for genetic counseling. And the allele specific oligonucleotide, they are specific alleles of a cell it recognizes, and it is used for genetic counseling and DNA fingerprinting. Then you have the highly repetitive sequence probe. These recognizes the repetitive sequence, that is, we say the variable number of tandem repeats, or we call it as VNTR, and this is useful in DNA fingerprinting. So here is a flowchart that shows how the DNA probe functions. The double-stranded DNA is there by heating or by action of an alkali. The single-stranded DNA are constructed. These are labeled with a probe, that is either a DNA or an RNA, and then we get a probe hybridized with a complementary sequence of DNA. Okay, so this is about DNA probe. So moving on to another short answer that can come, that is DNA microarray analysis. Okay, so this technique, it is used for the study of genomics of cells and organisms and a specific tissue under the various physiological and pathological condition. It is used to study the expression of a large number of genes simultaneously. So that's the importance of DNA microarray analysis. So there is principle of microarray. It is based on the nucleic acid hybridization. The technique uses DNA microarray or DNA chip, and it consists of a solid impermeable surface such as a glass slide on which hundreds to thousands of the DNA probe are attached, added by a computer assisted technique as micro drops, and these are fixed to support surface by chemicals, UV radiation, etc. Then the DNA or the RNA to be analyzed is labeled with radioactive isotope or fluorescent dye, and then the reactions are deducted by label and quantified by high resolution imaging and by computer analysis. So this is going to be the principle of the DNA microarray. So the size of the chip that can be varying from 1 into 10 into 5 micrometers to 100 to 200 into 20 micrometers. So there are thousands of oligonucleotide probes, each complementary to the specific gene, which are immobilized. A single chip microarray for human genome is being developed that will contain 1 million oligonucleotide probes. So in the next slide, you can see how this microarray is done. So you have the mRNA, and then you have the reverse transcriptase by which the DNA is synthesized. This mRNA, then it is denatured, and then application on the DNA chip. So this is the DNA chip we can see. So what happens here? After that, the hybridization with the fluorescent labeled DNA or the RNA, and you can find that there is the fragments that you can see that is analyzed or the microarray. So what are the applications of the DNA microarray analysis? It is used, it allows a study of a whole genome of a cell or an organism under different physiological and pathological conditions. A simple microarray analysis generated over a time course can indicate the coordinated genes involved in expression. It also helps in detection of gene causing inflammatory diseases, and it also helps in detection and screening of single nucleotide polymorphism. So this is one answer for DNA microarray, or it can also come as DNA chip. So moving on to the same short note concept, so we'll go on to another short note that can be asked, that is DNA sequencing. So DNA sequencing can be done by two methods, by Maxim and Gilbert's method, and then you have the interrupted enzymatic cleavage method or the deoxynucleotide method by Sanghas method. Then you have the automated DNA sequencing, and then you have the DNA chip or the microarray method that we've already discussed. So moving on to the DNA sequence, the first one, the Maxim and Gilbert method. So this Maxim and Gilbert method. So the first step here is the DNA strand is labeled at one end with a radioisotope. The two strands of the DNA are separated and labeled. DNA is distributed across four separate tubes. Each sample is treated with a chemical that specifically destroys one of the bases. It can be either the G or the C or both the bases, like A plus C or T plus C in the DNA sequence. Okay, so as a result of this, the DNA strand of the partially digested in the four samples at sites G, A plus C and T plus C and C. So the multiple label fragments of the different links are formed. The actual length of the fragment depends on the site at which the base is destroyed from the labeled, and then the labeled DNA is obtained. So the fragments obtained in four tubes are subjected to electrophoresis and then they are deducted by autoradiograph. The sequence of the bases in the DNA can be determined based on the bands on the electrophoresis pattern. So you can see here the double stranded DNA. So the single stranded DNA is obtained and it is labeled and it is distributed into the four test tubes. As you can see, the G base, A plus G, T plus C and C. So the specific bases destroyed and the fragments are formed. The fragments are separated by gel electrophoresis. As you can see here, the pattern, electrophoretic pattern, which can be seen. So on the bands on the autoradiograph you can obtain, and this by electrophoresis, the autoradiograph bands you can see, and whichever is specific, we can look for the specific nucleotide base pairs. Okay, so this is the Maxim and Gilbert's method. Next, you have the Sanghas method. Okay, so this is the interrupted enzymatic cleavage method or the dideoxynucleotide method by the Sanghas method. So this based on the random termination of the DNA chain during the enzymatic synthesis. The technique is possible because the dideoxynucleotide analog of each of the four normal nucleotide can be incorporated into the growing DNA chain by the DNA polymerase enzyme. So the ribose of the dideoxynucleotide has a hydroxyl group at both the ends, that is in the 2' and also in the 3' position, replaced with a proton, whereas the dideoxynucleotide has only a single hydroxyl group. It is replaced by a proton at the 2' position. So what happens then? The dideoxynucleotide incorporated into the growing chain is unable to form a phosphodiester bond with another dinucleotide base because the 3' position of the ribose does not contain a hydroxyl group because it contains a proton. So the growing DNA molecule can be terminated at random points from the first nucleotide to the last by including in the reaction system both the normal nucleotide and the dideoxynucleotides at such concentrations that the two nucleotides compete for the insertion into the DNA strand. So you can see in the next slide how this. So this is the DNA template, the primer that contains the hydroxyl group. It is treated by the dinucleotide bases, and you can see here there is the strands which are obtained, which are of different length. This is the 3' end, and then you can see the 4 dideoxynucleotide bases that are added, and then you can see the radioactive fluorescent dye that is inserted. You can see in this each of the nucleotides you can see. Okay, then what happens after that? These are subjected to the autoradiograph and you can see the separated strands, and there you can see the final sequence, that is from the 5' end to the 3' end, and then here you can see from the 5' end to the 3' end the required DNA fragment to be analyzed. Okay, so this is the Sanghas method. Then the last method you have is the DNA sequencing or the automated DNA sequencing. So the DNA sequencing is carried out by an automated DNA sequencer. The fluorescent tags are attached to the chain terminating nucleotides. This tag terminates the new strand synthesized by inserting itself into the DNA molecule. So four different new fluorescent dyes are used to identify the chain terminating reaction in a sequencing gel. The DNA bands are separated by electrophoresis and detected by their fluorescence. Okay, so this is a very accurate technique with high speed, and you can obtain it as early as possible, and has to be used, can be used for a it can be used for human genome project also, and for the gene therapy. And most important, the DNA sequencing methods can be used to direct sequencing to determine any mutation in the gene, and it can be used to detect any of the genetic disorders, for example hemophilia or B, which can be identified. Okay, so these are the applications of automated DNA sequencing, right? So this finishes with certain topics of molecular biology which can come as short note. In the next class we'll finish with some more topics which we have to write as a short note in molecular biology, right? So thank you, have a nice day.